Hello and welcome to this video on synthetic biology and specifically designer genes. Synthetic biology is a broadly defined combination of scientific disciplines ranging from the fundamentals of biology to molecular biology, all the way to electrical and computer engineering. It is a process of recreating life as we know it using means created by humans. This creation can be used as part of new parts or systems in cells and organs. At this stage, we're not quite at a bionic human, but certainly augmentation or supported systems. This includes the previously described organs on a chip, designer genes, proteins, and even the beginnings of artificial nucleotides. The most extreme example of this is the audacious work of Venter in 2010, where he and his team created a synthetic replica of Mycoplasma microides. Of the many developments in synthetic biology, this video will focus on designer genes. For the purposes of this video, designer genes fall into two broad categories: research tools, and arguably as therapeutic or medicinal goods. Although the overlap is substantial, there are some clear differences. You can consider designer genes as something along the lines of PCR primers. Conversely, when you consider medical tools, the best example is a relatively recent gene therapy for Duchenne muscular dystrophy called epilersen. Let's begin by examining research tools, which can be better isolated and described. The first, PCR primers. These work on a specific gene by matching the RNA product in cells, serum, or other samples. These RNA sequences are in circulation because they are the gene that is being expressed. The RNA is then backwards processed to create what is known as a complementary DNA strand. This is for most intents and purposes an accurate copy of the gene. Through the PCR process, this gene is repeatedly copied. Now the key to creating this designer gene is actually in the very early stages: custom PCR primers. These both are and need to be custom made to fit the gene with exacting detail. For the purposes of this video, let's assume the exact details are unnecessary, but bear in mind that you need an exact match for the nucleotides, guanine thymine weight, temperatures, and more. You can select, modify, and otherwise change the primer you use to make this selectivity happen with varying degrees of success. Now that you have a gene in a large quantity, you can create plasmids and other vectors to deliver genes. You can see an example of this in the linked videos, GMO yeast and FDA vs biohackers. As was highlighted in the latter, this technology is important when developing GMO research animals. The video highlighted how specific hereditary defects caused by mutation were being corrected using GMO technology, but more on that later. The strengths of this approach are myriad, but to specify some examples, there is price, convenience, reliability, specificity, and more. The weaknesses, although, are substantial as well. These are easily outweighed by the benefits. The specialized hardware is a significant drawback to this, as it is very expensive. It needs special preparation and can use incredibly toxic reagents. The biggest hindrance with this is toxicity, which often makes it a destructive process, and if not destructive, limited to small cell samples such as stem, oocyte, and prospectively cancer cells from modelized lines. The extreme version of these plasmids are the artificial chromosomes. The example of this linked in the below description box explains how 500 modifications were made. This involved 273,000 base pairs knitted together, and that they had taken out 53,000 base pairs. This made it shorter than the original. The cell that received this streamlined DNA survived and could be propagated. To know something of the research used for designer genes, let's look at medical applications. Many conditions can trace a fundamental cause to a genetic defect, whether that is hereditary or acquired during life. If medicine can provide a treatment, pharmaceutical companies will try to satisfy that market. A recent example of this involves a clinical trial for epilersen. This is a synthetically created gene for Duchenne muscular dystrophy. Muscular dystrophy is a cluster of disorders caused by a large range of mutations in a wide range of genes. The treatment described here works by distributing a replacement gene across all the muscles in the body. Once there, it substitutes the native gene and a functional product is expressed. This may not seem like much, but it is a significant step when you begin to break down the components that make this drug work. First, the company which makes epilersen is not using the full gene, but a small, simplified version. This, of course, has a direct effect on the complexity and size of the dystrophin protein. The reason for this is the capacity of the vector and carrier they used. It cannot carry the full gene and a vector which works. Therefore, the original gene would never have been an effective approach. The truncated gene that they did use, and as a consequence truncated protein, were expected to be more functional as an option, but worse in their functionality compared to the native gene. However, there was good news. The new protein had very good results, more so than they anticipated. It should be obvious that this is not a naturally occurring gene or protein that they found in another species, but something that was the development of the lab. The development of the gene and protein pivoted on the functionality and ability to deliver this gene. For this reason, significant modification was needed just for delivery. When you consider then function and the possibility of the gene causing immunological side effects, this was a major endeavor, as they now feel it's safe to proceed to clinical trials. It is fair to assume that they've not only been able to figure out a way around those immunological problems, but to find something that is functional enough that it can be used to treat this disorder. This is just one example of how a synthetic gene or a designer gene can be used in medical applications. There is a final area of interest, CRISPR technology, which has successfully been used in embryo editing in the last few weeks, and more widely in research. An example of this is for sickle cell anemia. In doing so, there is a crossover between the medical application and research applications. In the case of embryo editing, CRISPR was used in the first few days of development in a small number of cells, well under 100 and possibly as few as 1. Nominally, this would continue to propagate and form a blastocyst with about 200 cells. These will normally form into a ball. That is under normal circumstances, but in this study, scientists used gene editing to remove something called the OCT4 gene. This gene is essential to forming a blastocyst, and from a blastocyst, eventually a fetus. This is an application and a technology which takes CRISPR from being a sharp knife to a surgically precise scalpel. This technology has also enabled researchers and IVF labs to look at correcting gene abnormalities before birth. In the article linked below, the researchers used gene editing to remove the genetic mutations responsible for a connective tissue abnormality. If untreated, the embryo would never be able to develop. At the moment, this technology is limited in how effective it is, with high-profile examples having success rates between 7 and 88%. This wide range of possible targets and possible success is simply unacceptable for anything being used in humans. Other trials have examined the use of CRISPR to correct heart defects. Conversely, the same technology can be used to create genetic and anatomical defects in research animals. These can be passed on to future generations. This creates a viable means of humanely altering the genes of research animals and creating models of disease without the need for surgical, pharmaceutical, or environmental methods that could be considered cruel and unusual. In this respect, this approach has both a medical treatment aspect and it has a research aspect. The two can both go hand in hand or can be diametrically opposed. These three examples of designer genes give you an understanding of how custom genes are used in research, medicine, and in between. It does raise questions of ethics, morality, and the future. The next video looking at will examine artificial nucleotides, which are the building blocks of life. Thank you for watching this video. If it has been of interest, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.